Why don't we go ahead and get started? Thank you everyone for joining. It's a pleasure to be here today for Shersingh's talk and then his defense with the committee immediately afterwards. My name is Rob Jackson. I'm a professor in Earth System Science. I've had the pleasure of working with Shersingh for the last years. And he came to us from the University of New Hampshire. He was a presidential honor scholar there with degrees in conservation, sustainability, and forestry. And he perhaps first came to Stanford campus as part of our SURGE program, the summer undergraduate research program. I believe worked with Paula Wielander and Scott Fendorf, if I'm not mistaken, but it brought him to Stanford for that summer. And then we were lucky enough to entice him to come to graduate school here, where he joined my lab for a PhD. I'll give you just a very short background and some of the reasons I'm really excited to hear his talk today and why it's been a pleasure to work with him over the past years. And also I'll show you a few very quick pictures very briefly. Shersingh is a first-rate ecologist. Since being at Stanford, he received a number of fellowships that are prestigious, National Science Foundation, Ford Foundation, McNair Scholars, Udall Foundation, all in his time at Stanford. Even more importantly than his honors, he is our lab chef, and just brings a lot of brings not only science into our lives but a lot of joy into the lab group and into the community and the department as well. So he has also, I think, made me and the Stanford community better through his commitment to issues of diversity, equity, and inclusion. As many of you know, Shersingh volunteered in programs like Canopy, which is an urban forestry program, and worked with folks in East Palo Alto. He volunteered in the GeoKids program, teaching science to elementary school kids, and lots of other things. And he also organized the Conciencia seminar series, which brings Earth and ecological sciences to Spanish-speaking audiences, and he still continues to do that. So he's been really committed to these issues, and I really think that it's been wonderful to see that commitment and how much he's gotten done in that space while finishing his PhD. So let me show you a couple of pictures. Nothing too embarrassing, but I'm assuming that you can see that. I want to acknowledge Hannah too. A PhD and a graduate degree is never a one-person endeavor, so it takes commitment from a couple in this case. And in Shersingh and Hannah's case, we all know that their dogs are a big part of their life too. So I acknowledge their dogs as well. There are a few pictures. I believe the upper left photo is in Point Reyes, was an outing. You see a few other lab members there. You see Shersingh's legendary hot sauce in the lower left photo that he brings each year when pepper season arrives, which is upon us now. And you don't have to fish in my lab to graduate, but it helps. So Shersingh has a lot of hobbies and does a lot of things in addition to his science, which is important to maintain that work-life balance. Some years ago, he gave a talk on what he wanted to be when he grew up. I'm not sure if that's a firefighting outfit, but I don't know how you answered that question, Shersingh, so many years ago, but I hope it was to be an ecologist working in your garage during COVID, because the labs are all closed. So Shersingh's worked tirelessly to finish in difficult conditions, as others on this call have too, and COVID hasn't made it easy. So I just want to acknowledge your work and your passion for science and your passion for life, and looking forward to hearing your talk. So take it away, Shersingh, if you will.
Thanks, Rob. I really appreciate that. It means a lot to that introduction really means a lot. It's been a huge privilege for me to be part of your lab and part of Stanford. So I am going to start sharing my screen. Okay, can everyone see that all right?
Yes.
Okay, thanks, Colin. Okay, Well, yeah, thanks everyone for taking the time to learn about my research. Like I said, I feel incredibly honored and fortunate to share the space with you all. I also recognize I've been afforded a lot of privileges during my educational journey at Stanford and at the University of New Hampshire. And I'd like to start by acknowledging that I've spent most of my lifetime on the ancestral and unceded lands of the Wudinkan Taino, the Nipmuc, the Abenaki, the Muwekma Ohlone, and the lands of many other native people who stewarded the land prior to European colonization. And I also want to acknowledge the hardships that Indigenous people continue to face to this day, and the responsibility that we and the institutions we're a part of have to foster the well-being of those that came before us. So as I shift into my research, I'm sure at least half of the audience is wondering why I decided to do a PhD on the size and shape of plant root systems. And for me, as a terrestrial ecologist, my principal goal is to understand some of the biggest questions in ecology. And some of those questions really lie beneath the ground. And there are many reasons for that. But first, it's important to acknowledge that roots significantly impact the Earth system and our understanding of the Earth system, especially in the ways that they move water, carbon, and nutrients throughout the environment. And a recent study even found that plant root systems make up 25% of overall plant biomass, which means they're a significant portion of the global carbon cycle. And to really show the way that they impact the environment, I wanted to show this, this figure that shows the difference between two different rooting models, this dynamic root model and this control root model. They're basically just two ways that people have predicted the size of plant root systems. And when we look at the difference between them, which is shown in deep red colors and the darker blue colors, we see that there are parts of the globe where just changing the distribution of plant roots can really affect our understanding of things such as gross primary productivity, which is essentially how much stuff plants make and through photosynthesis. But it's not just, roots don't just affect the growth of plants. They also interact with how much carbon our ecosystems hold, the nitrogen uptake, and the evapotranspiration. So in many parts of the globe where, again, you can see the dark red colors and the dark blue colors, just by shifting our understanding of the size and shape of plant root systems, it can really affect our predictions of how climate change or how the Earth system works. And part of the reason for this is that the data that we need to really understand the size and shape of root systems is really lacking at the global scale. For example, some of the largest plant trait databases, such as the GROOT database, the TRI, and the FRED database, have all stated that we just don't have the data necessary to understand the depth and spread of plant root systems. Two very basic questions: how deep plants grow and how wide they grow. To go into a little deeper, the TRI database showed that 10% of all of their thousands of requests are for max rooting depth data. But there's actually one of the traits that they have the least amount of coverage for, or at least relatively less coverage for. And what we hope with the work I've done is to try to fill some of the gaps on understanding the size and shape of root systems at the global scale. And to go a little further as to why plant root systems are so difficult to understand and estimate, one of the reasons is that plant roots interact with so many different parts of the environment. The size and shape of root systems is affected by the location of water and nutrients below ground, and other soil properties such as depth and texture. And overall, the below ground environment is just a very complex place to work. Roots are also incredibly difficult to measure and observe. There's a really good understanding of above ground dynamics, partially due to the fact that there's been decades of systematic sampling and inventories to understand how large plants are above ground, especially in sectors such as forestry, where the main product is above ground biomass. Also, we can use information, or we can use techniques such as remote sensing from satellites, drones, and even simple cameras to monitor the above ground parts of plants. However, it's a lot more difficult to understand what's happening below ground. Often you have to dig up roots or dig holes to understand what's happening. And I think this cartoon really sums it up, where I appear to be looking at the canopy of plants with a little bit of anger and maybe a little bit of jealousy, just wishing that roots were that easy to understand. And for these reasons, my goal was to find different ways to understand the size and shape of plant root systems. And kind of my research in a nutshell, I want to understand how big are plant root systems. And in chapter 1, the main question is, how deep and wide are plant root systems? And how does this shift across climates? In chapter 2, I go a little further to try to understand the amount of space taken up by plant root systems. And is there a relationship between the size of plants below ground and the size of plants above ground? And in chapter 3, we go to a case study where we look at the more local effects of plant root systems, such as competition, water availability, and nitrogen, to see how these factors affect the depth of and distribution of plant root systems. And again, to look at all these things, we developed different tools and techniques throughout my PhD. I approached some of these questions from, instead of focusing on just one tool throughout my PhD, I wanted to figure out how I can use all our available data to understand some basic information on the size and shape of plants. This includes tools such as big data synthesis, machine learning, developing image analysis pipelines, stable isotope ecology, among many others. So now I'm going to shift back to chapter 1, where the focus is understanding the size and width of plant root systems and their interactions with climate. And I want to mention that this chapter, we have submitted it to new phytologists, and it's currently in the revision stage. So previous work on understanding the size and shape of plant root systems have shown that there are relatively deeper roots in more arid and seasonally dry climates. For example, a study by Fan et al. Found that if you understand the variation in the soil water profile, you can understand a lot of the variance in the rooting depth of plants. And the study by Shersingh and Jackson in 2005 really summed it up well, where they were able to predict the probability of deep rooting based mostly on climate. And they found that in seasonally dry and arid climates, such as savanna and the cerrado or monsoonal forest, you're more likely to have deeper roots, but then there are shallower roots in boreal, Arctic, and climates that are ecosystems that have annual plants. So generally, in dry and more seasonal places, there are deeper roots. In colder, less seasonal, and wetter places, there tend to be shallower roots. But we wanted to go further by understanding how the dimensions of root systems relate to the size of the plant above ground. So the first question is, which factors influence those plant dimensions across the globe? And do the dimensions shift across climate? And when I talk about the plant dimensions, I'm specifically referring to rooting depth, which is the maximum depth reached by plant root systems, the lateral spread, which is basically the radius of a root system. So how far from the plant does a root system grow? The shoot height, which is the height of the plant, and the shoot width, which is how wide a plant is above ground. So these four are the main dimensions I'll be referring to throughout this chapter. And to investigate that, we created a new version of the root systems of individual plants database. And this database was first published by Shersingh and Jackson in 2002, mostly for more arid and seasonally dry climates. It was later expanded in 2005 to include around 2,500 observations. And as part of my PhD research, I've expanded this database to over 5,000 observations, more than doubling its previous size. And this now includes information on plant dimensions, but also a lot more information about the ecosystem and the climate that these plants are grown in. And you may be wondering, so where does all this data come from? The data we collected came from the literature from over 609 different studies that all showed the maximum rooting depth of plants. And to observe the rooting depth of a plant is a ton of work. It is incredibly time intensive and costly. For example, on the left, you can see this apple tree that was carefully excavated, almost the same way you would excavate the bones of a dinosaur, in order to demonstrate the distribution of the root system. And it was hung up on these cables. And even for much smaller plants, it's still no easy task. On the right, you have a scientist in a soil trench, and he's using a knitting needle to untangle the roots of individual grass species. And that way they can look at the size and shape of those roots as well. So our goal was really to take advantage of all of this previous work, and this has spanned over a century of different ecological studies. Here I wanted to show just another map of our data that shows the distribution a little bit more clearly. So this is a hexagonal binning where the yellower colors represent places where we have the most data. So places like Central Europe and the Southwestern US. However, for the most part, we have a pretty good coverage of a lot of the global ecosystems. And this comes from observations from 3,000 different species from 200 plant families. And if we look at that across the climate, on the y-axis, we have mean annual potential evapotranspiration. You can think of that like temperature, essentially. And here we have precipitation. This gray background is every climate on land. So the main point is that we have a really good distribution of observations across a lot of the climate space. Potentially, the places where there should be more investigations on rooting depth would be the most humid climates that would be represented by the areas out here. So now that I've summarized where the data comes from and what the data looks like, now I'm going to shift into looking at the overall trends in plant root system size and shape. So first, woody plants make up the deepest rooting depths. On the y-axis here, we have the max rooting depth, and we see that smaller plants like forbs, grasses, and succulents have some of the shallower rooting depths, followed by semi-shrubs, and then woody plants have the deepest roots. And the deepest known root system actually grows all the way down to 120 meters in depth. And if we look at the width of the root system, the lateral spread, again, it's at the farthest distance reached from the stem of a plant. Woody plants, again, the trees and shrubs, have some of the widest root systems. But actually, succulents now have wider root systems than forbs and grasses, meaning that succulents overall have kind of short, wide root systems. But you might be wondering, so where is there a tree that roots all the way down to 120 meters? So the deepest root system was actually found in this wild fig tree in South Africa. And the largest lateral spread, again, that width from the center of the plant, was found in a balsam tree in Namibia. So both in Southern Africa, in seasonal dry climates. But to put that into perspective, if the Stanford tree had the world's largest root system, its root system would be the width and the depth of the Stanford Stadium football field. But how does the size of root systems relate to the size of plants above ground? We found a really strong correlation between the size of plants below ground and the size of plants above ground, which means that the depth and spread of root systems co-vary strongly with the height and width of plants above ground. For example, here we have maximum rooting depth on the y-axis and the size of the plant above ground on the x-axis. The different dots going from lighter to black are grasses, forbs, and woody plants. And the main takeaway is that no matter the size of the plant above ground, they tend to scale, the rooting depth tends to scale in relation to overall plant size. And we see the similar relationship for lateral spread. Now, lateral spread, again, is the width of the root system. And there's even a stronger relationship at the global scale between the size of a plant above ground and the lateral spread, which could mean that lateral spread might be an important factor in terms of anchoring a plant in place. So root systems need to grow wider in order to keep a plant in place, the larger it is. And a general trend we found is that plants are 2¼ times wider below ground compared to above ground. So when you're walking around and looking at plants and you see how big the tree is above ground, just imagine how much bigger that plant must be below ground. And if you're like me and you're walking around in the city, you might feel bad for trees in small little sidewalk boxes. But another thing we've seen is that herbs and annual plants that tend to be much smaller below ground can take up a large amount of space below ground. But the next question is, how do these factors shift across climate? So what we did was we implemented a machine learning approach called random forests to understand which factors in the climate and environment most affect the size and dimensions of plants. And what we found is for max rooting depth, which is the depth of the root system, metrics such as metrics that relate to temperature seasonality, such as temperature of the driest quarter of the year, were among the most important in terms of predicting the maximum rooting depth. But for the lateral spread, again, temperatures relating to the size of the plant above ground, so shoot height and shoot width, which is how tall and how wide the plant is above ground, were the most important. So there's a difference in how these plants scale in relation to different climate and environment predictors. But if we look at the plant then above ground, so shoot height and shoot width, these two are more related to factors of plant physiology, such as growth form and family, as shown here. So max rooting depth is more affected by things such as climate. The width is more affected by the size of the plant above ground, but above ground, the size is more constrained by physiology. And we also found that the dimensions of plants then tended to shift across climate. So in this figure, we have an axis from arid climates all the way to humid climates. And the y-axis represents the dimensions of plants, where a number that is below this dashed line, the 1-to-1 line, is a plant that is wider in relation to its height. And a number above that is a plant that is taller in relation to its width. So as shoots go from arid to humid climates, they go from wider to taller across the globe. And if we look at the plants below ground, they go from deep and narrow and arid climates to widen shallower and humid climates. So there's what we're seeing is a divergence in the shape of plants across climates. And just to show some of the takeaways for chapter 1, we created, to our knowledge, the largest database of plant dimensions across the globe. And we hope that this data can then help further our understanding of the Earth system and the way that roots interact with climate and the environment. We saw considerable variation in plant size, where most roots don't root deeper than 75% of roots don't root deeper than 2 meters, but many roots can also root as deep as tens of meters. However, roots tended to go very strongly with the size of plants above ground, especially the lateral spread. And plants tended to be 2¼ times wider below ground than they are above ground, with deeper and narrower roots, with there being deeper and narrower roots in more arid and seasonally dry climates. So we hope that these findings can really help push our understanding of the size and shape of plant root systems. And for chapter 1, I'd like to acknowledge Rob and Jochen for starting that RSIP and all the people that helped put the data together and everyone that's published the data in the past. So now I'm going to shift to my second chapter, which is focusing on the above to below ground relationships of the size of plants. And this is a continuation of chapter 1, where we saw that the size of plants covaried strongly with the size of shoots. If you remember this graph, we saw that the width of plants related very strongly with the width of shoots. But in chapter 2, we want to go further by understanding how the volume of space taken up by plants and the biomass of plants, we want to understand the above to below ground relationships of the volume and biomass. And I do this using allometry. And allometry is the study of how body parts scale with overall body size and with each other, and the impacts that it has on ecology and evolution. And one of the first examples of allometry was by Huxley in 1924, where they found that the right claw of this Chelea crab scales to the 1.5 power of the body size. So as the crab gets bigger, its claw grows at a faster rate. So that's kind of one of the first known examples of allometry. And most allometric models kind of follow this basic relationship where the size of body part Y equals the size of body part X to this exponent times a beta coefficient. And in this chapter, I'm going to be mostly focusing on understanding this alpha coefficient. Another example we might be more familiar with is the size and shape of humans. So here we have the size of our body and the size of an organ. And the idea is that as the body size gets bigger, the size of our heart scales proportionally to the size of our body. However, the size of our brain scales differently. From age 0 to age 6, the size of our brain scales at an alpha of 0.75, the size of our body. And these relationships are really helpful to understand biology and evolutionary history. When scaling happens with this alpha of 1, we call this isometry, and I'll be referring to isometry throughout. So isometry is a relationship we saw with the heart to the body, for example. And one of the goals in the field of allometry is to find a universal theory. Is there one universal theory that predicts how different organs scale in relation to each other? And no theory has been more impactful than the West Brown-Enquist theory, where they predicted these 1/4 scaling relationships. And the West Brown-Enquist theory relies on the following assumptions. The first is that the cross-sectional areas of branching networks should add up to each other. So if you think of something like a Twizzler or a string cheese, the more you rip apart the Twizzler or the string cheese, those smaller pieces will always add up to the size of the original bigger piece. And most biology is made up of branching networks that support the plants and animals. And the growth of any plant animal is limited by the capacity of that branching network. So these are some of the basic assumptions in allometry. Some examples that we may be familiar with, again, are our circulatory system, like the heart, where the heart can only needs to be big enough to support pumping all the blood we need in our bodies, or your lungs, the branching networks of your lungs and your respiratory system needs to be large enough to provide enough oxygen. But even things like sewer pipes scale according to this theory, where the size of your main sewer pipe needs to be large enough to support all the sewage coming from individual houses. You can't add on more houses or create a bigger city without increasing the size of that network. So these all fall under the field of allometry. And these same relationships apply to plants. So above to below ground volume and biomass of plants should scale isometrically. And isometrically, again, means with that exponent of 1. And this is based on the assumptions that the size of the cross-sectional areas of stems and roots are equivalent. Basically, the pipes of roots and stems should be equivalent due to the fact that plants need to conserve the flow of water from the roots to the stems and out to the leaves. And plants throughout evolutionary history have optimized the size of their tissues in order to make the flow of water more efficient. So again, the idea is that the above ground size of plants should scale proportionally to the below ground size of plants. And this symbol, I'll be using it in this chapter, and this symbol essentially just means that it means proportionally. And one of the studies that tested this allometric relationship of above ground size being proportional to below ground size was published by Brian Inquist and Carl Nicholas. And they found here on the y-axis is above ground size, and the x-axis is below ground size. They found that this relationship was isometric. So the size of the plants scaled proportional to each other above and below ground. This alpha was equal to 1, and that means isometric. But, you know, I've spent a lot of time introducing what allometry is, and it might sound a little bit complicated, but there's a lot of reasons why we would want to understand how organs scale in relation to each other. If you remember this is a lot of work, and it would be a lot easier if we had well-established relationships to predict the below ground size of plants just by using data on what we know about plants above ground. And we can use all of this backbreaking work to excavate and measure plants to create allometric models and test the theory behind allometry. And beyond making it just easier to understand the size shapes of plants, it will provide valuable insight on the biology and the evolutionary history of plants and the ontogeny of growth. Furthermore, understanding the location of roots is really valuable. Biomass is nothing about the spatial relationship of a root system or where they are. And past studies have mostly focused on understanding this ratio of above ground biomass to below ground biomass, normally known as the root to shoot ratio. However, knowing the biomass of a plant doesn't provide information on the location, and to understand the movement of water, carbon, and nutrients from below ground to above ground, we need to know where those roots are. But there's almost zero data on the volume of plants. An example of this is a search of the Fine Root database showed that there are only 86 observations of plant volume, all for one tree from one study. One of the opportunities, though, is to use all of the current biomass data to figure out if there's a relationship between biomass allometry and volume allometry. So our goal is to understand if the volume of soil occupied by the root system scales proportionally to the volume of space occupied by a plant above ground, and if volume scales similar to biomass. Because if volume does scale similar to biomass, we can use all the data and knowledge we have about the biomass relationships to say something about the volume of plants. And the big question is, how did I create data on this? If you remember, I said there's very little information on the volume of plants, which is why there haven't been these volumetric relationships developed yet. And so we needed to develop this data ourselves. And we use this series of books called the Wurzel Atlas, created by Dr. Laura Kuchera. And this spans now it's actually over 60 years of work. It was published in the 1960s and is a series of plant profiles of showing the depth and distribution of plant root systems. For example, here we have one of the profiles, and each of the 1,500 profiles in the Wurzel Atlas contains information on the plant, the scale, the location, elevation, and a really detailed soil description, all in German. And even the width of the roots in the profiles were drawn to scale with using pencils of various widths. From the Wurzel Atlas, we pulled out 206 tree profiles. took each of these drawings from the books, we digitized them and recorded information on the plant. We ran them through a machine learning thresholding process and then manually traced the remainder of the images. We then developed an image analysis pipeline with the help of Guillaume Lebay, who provided the RIA software. And from the RIA software, we wrote code to measure volume and other plant geometric traits. And we combined all the output from our image analysis to the original data from the books. And as I mentioned, from RIA, we're able to measure a variety of traits such as the stem length, root length, the volume of the tissues themselves, and the vertical distribution of plants. But my results here will really focus on system volume, which is the volume of space occupied by the root system or the canopy. And to do this, we essentially traced around the outside of the roots and the stems, and then we rotated that tracing to create an estimate of the amount of space taken up by the root systems. And this resulted in 206 profiles of plant volume from a variety of trees. And here we can see a phylogenetic tree showing the evolutionary history of the plants, with the purple being gymnosperms and the black observations being angiosperms. And this is just to show that we're able to cover a wide variety of plant types from different plant families. From this volume data, we connected it to biomass data from the biomass and allometry database. We took it's called the BAD database. We took the BAD database and pulled out only observations that matched our volume data in order to have both volume and biomass for the same types of trees. Now, I mentioned two different types of plants, gymnosperms and angiosperms. Well, angiosperms are plants that have enclosed seeds or flowering plants. So you can think of any fruit or vegetable you've ever eaten is probably an angiosperm. Other examples are apples, oaks, anything with a flower, grasses and forbs. And gymnosperms are an ancient type of plant growth, much simpler type of plant. And conifers and ginkgos fall within this. So we separate our data into angiosperms and gymnosperms because previous studies have shown that their biomass scaling relationships are different from each other. So we would predict that the volume would probably be different from each other too. So here is the data on the volume of space taken up by the root system to the volume of space taken up by the canopy. And we predict that this relationship should have an alpha of 1, so it should be an isometric relationship. And for angiosperms, what we found is that the volume alpha is less than an alpha of 1, whereas biomass is nearly isometric, nearly an alpha of 1. So what does this mean? If we have the canopy size and volume here and the root system volume and biomass on the y-axis, it means that as the plant gets bigger, the volume of the shoot grows proportionally larger in relation to the volume of the root, but the biomass relationship is relatively linear, so it has an alpha of 1. And the biomass is the yellow, the volume is the blue. If we shift and look at gymnosperms, both the volume and the biomass have an alpha of 0.9. This means that volume and biomass scale at the same rate as each other. And you could potentially use a lot of the data we have on biomass to help predict the volumetric relationships of gymnosperms because they have a similar alpha. And you can see on this line here that while the curves are not right on top of each other, they have a similar slope. But what does this mean in terms of the overall above to below ground relationship of plants? For gymnosperms, which are the last example I showed, the volume of space taken up by the plant, a majority of the plant volume is below ground. Excuse me. Where an average of 84% of plant volume for gymnosperms is below ground, compared to most of the biomass for gymnosperms being above ground. For angiosperms, we see a similar relationship again, where angiosperms tend to take up much more volume below ground and more biomass above ground. Overall, 86% of the space occupied by trees, by space, I mean the 86% of the volume occupied by trees is below ground, compared to only 22% of biomass for gymnosperms or 41% of biomass for angiosperms. This means potentially that using the biomass relationships, the root-to-shoot relationships, to predict volume might lead to an underestimation of the size and shape of plant root systems. So biomass is really good at helping us understand carbon dynamics, but it's not as helpful when we're trying to predict where roots are, and that's important for understanding the uptake of resources, the location of roots, and rhizosphere processes such as plant-soil interactions and plant-biota interactions. And using biomass root-to-shoot relationships could lead to an underestimation of root system size, because as we noted before, the volume occupied by most of plant volume is below ground, whereas most plant biomass is above ground. So overall, for chapter 2, we created a new way to look at the volume of space occupied by plants, and found that the volume and biomass of gymnosperms tend to scale at similar rates, and the biomass of angiosperms tends to scale nearly isometrically. But 86% of the space occupied by trees is below ground. So I'd like to pause and just acknowledge all the people who helped with the image tracing, with the development of the image analysis pipeline, and people who provided biomass data as well. But especially Dr. Laura Kuchera, who spent basically her entire career developing this Wurzel Atlas. So in chapters 1 and 2, I spent most of my focus was on understanding big data analyses at the global scale, specifically looking at the dimensions of plants and rooting depth. And I showed that the size of rooting depth is affected by climate and also is affected by the size of plants above ground. However, at the global scale, we're not able to measure any of the hyper-local effects on plant root systems, such as the way plants are affected by competition, plant-soil interactions, such as the availability and limitations of resources such as nitrogen, or water availability. So overall, it's really difficult to understand these local effects from using these local effects using global studies. And that brings us to chapter 3, where we're looking at the root system distribution and resource uptake strategies of co-dominant trees in a northern temperate forest. And to do this, we zoomed in from this global perspective to a more local perspective. And specifically, we zoomed into Thompson Farm, which we use as a case study. Thompson Farm is part of the University of New Hampshire in Durham, New Hampshire, and it's an experimental field station. It's a northern temperate forest. You can see actually a picture here of the canopy during the fall. That was taken from the top of this walk-up tower at the laboratory. And this forest is co-dominated by maples, oaks, and pines. This shows just the distribution of trees in our subplots. The height here is the height of the tree, whereas the width of the stems is the diameter of the trees. And the top of the canopy is mostly dominated by pines, followed by the maples and oaks, and there's a really dense understory of other hardwoods such as American beech. And you might be wondering, so why'd you have to go to New Hampshire? Aren't there forests somewhere between Stanford and the East Coast, right? Well, one of the reasons is that the species distribution at these sites really helped us look at the effects of competition. Another reason is that the shared facilities allowed us to look, have further collaborations, because the people at Thompson Farm have been able to look at many different parts of the forest dynamics. Furthermore, and this is the most important point, the northern temperate forests are undergoing severe change due to more frequent and severe droughts, and also due to effects in land use. 150 years ago, if you looked at the forests in New England, they were mostly deforested and were farms and fields. They were then abandoned, and the forest regrew from what used to be an oak pine forest to now a forest of more mesic species. But now that droughts are becoming more frequent and severe, there might be another turning point in the forest where they might shift towards more drought-tolerant trees, such as oaks. So Thompson Farm provides a really great opportunity to look at that due to the drought treatment plots at the farm. And when I mention these drought treatment plots, this is through the Northern Forest Drought Net network. And this experiment was developed and is led by Dr. Heidi S. Bjornsson at the University of New Hampshire. Here they built these roofs in the forest, and they basically are gutters that remove 50% of growing season precipitation. So 50% of the precipitation from the spring to the fall. And this simulates an extreme drought event, but it's an extreme drought event that's constant because they put up this experiment every year, and it's been continuously operated since 2015. You can see here a map of Thompson Farm, and these red squares are the drought treatment plots, and the blue squares are adjacent control plots. So using these drought treatments, I wanted to understand how the distribution of the roots of maples, oaks, and pines, if they differ from each other. Basically, do they have their roots in different soil depths to avoid competition? Also, does drought affect the vertical distribution of their root systems? Do plants and the trees shift their distribution, their rooting depth distribution towards deeper layers if they don't have water, for example? And at which depths do plants take up their water and nitrogen? So first, to look at the vertical distribution of maples, oaks, and pines. So we developed an experiment where we identified locations that had maple, oak, and pine within a 5-meter radius of a coring location. And you can see all the locations we sampled at in the white dots in the map above. We then cored using a power core down to 1 meter and took out the roots from the soil by sieving it, and we removed all the live roots that are maple, oak, and pine. The one big issue is that it's very difficult to do a visual identification of plant root systems. However, one of the main benefits of doing the study at Thompson Farm is that we are able to identify the roots of maples, oaks, and pines. For example, we have these images here published by Pregetzer et al. In 2002, where we see maple, oak, and pine. And then on the left, we have an image from our study. And so I want to give you all an opportunity to maybe guess which root you think, or which plant you think this root belongs to. So you can write in the chat, and I'll wait to see a couple answers if you can guess which root this plant belongs to. I'm getting a lot of pine from the audience. Okay, so sorry, I'm clicking back to Zoom here, to PowerPoint here. Yeah, so these roots belong to pine, and in pines you actually see a very characteristic forking pattern on a lot of their root systems, which kind of match what you see in this image below. So essentially, and actually after we do our visual root ID, we've sent our samples out to do DNA barcoding in order to confirm that our visual ID works, especially because this is a technique that's not often used. But the idea is we can ID roots the same way we ID leaves based on visual traits. Where maples have really long and skinny roots, they tend to have a yellowish hue and this characteristic bulbing pinching pattern in their roots. Oaks have a high branching intensity, so a lot of branches and a lot of root tips, and they have darker, redder hues to their roots. And pines tend to be really short and stubby roots, have a darker gray, black color and rougher bark or rougher exterior. And the reason pines are short and stubbier is because they tend to rely on other methods for resource uptake. So now I'm going to transition to showing what the rooting depth distribution of the trees look like in this forest. And we have separated the trees into maple, oak, and pine roots. The dashed line were the plants that were experiencing drought, that were under that roof in the forest. The solid line are in the control, so they were not experiencing that 50% reduction in precipitation. And what we found for the most part is that in both treatments, most of the root biomass was in the top 30 centimeters. However, now we're looking at the cumulative biomass, which means as you get deeper, we add up the overall amount of biomass. We saw that oaks had a major increase in the overall amount of root biomass in the drought sites. So the drought sites are the ones with the dashed lines and the triangles. So oaks had more roots when experiencing drought. Why would that be? Here I'm showing the soil moisture of the soil moisture content of both of these sites. The solid line is the control sites, did not experience the enhanced drought. The dashed line is the drought site. And we see that the soils of both of the drought sites were about had half as much soil moisture as the drought site. But overall, now the drought site in the dashed line had an overall increase in root biomass density, especially in shallow earth soil layers and in deeper soil layers. And a lot of this increase in root biomass was driven by that increase we saw from the oaks. And this could be potentially because the oaks are increasing their fine root biomass in order to take up more water. So oaks had an overall increase in the root biomass. But all of the plants actually had a shift in their rooting depth distribution. So what I'm showing here is the 50th percentile root threshold, which is the depth at which 50% of the roots are above and 50% of the roots are below. And I'm showing that here in this drawing for this plant. And for maple, oaks, and pines, that depth in the control sites was 3 to 5 centimeters. But in the drought site, we saw that the 50% depth for maple, oaks, and pines shifted by 80% towards deeper soil layers. And you can see for pines, it's really interesting because while pines did not have an increase in overall fine root biomass, their root profiles shifted much deeper from 5 centimeters all the way down to 15 centimeters from the control to the drought. So overall, we saw that water limitations led to relatively deeper root systems. And sometimes some of the plants have different strategies where oaks increased their root biomass overall, where pines mostly had a change in their root biomass distribution. So now we're going to shift looking at resource uptake. And this is because the location of roots might not indicate where they're taking up their resources. And we use this technique called stable isotope ecology, where we can sort of use stable isotopes as a fingerprint to understand where roots are taking up water and nitrogen. So how do we use stable isotopes? And this is a very simplified description of this. If we know, here we have a plant and we have the soil. Here is the ratio of nitrogen. So it's the ratio of the 2 stable isotopes of nitrogen, which occur naturally in the environment, and the ratio of 15N, which is 1 isotope of nitrogen, and 14N can shift for a variety of reasons. Now, if there's a gradient in the ratio of isotopes, we can use that known relationship to predict relative depths of uptake. For example, if the leaves of the tree had a ratio of, if we measure the ratio of 15N in the leaves of the tree, we can try to match that to the ratio in the soil and predict somewhat where plants are taking up their nutrients from. And that's what we did for nitrogen and for water isotopes. Again, this is a simplified approach to this technique. So to do this, we collected soil, water, and nitrogen from the soil. We collected tree nitrogen, which was collected using shotguns to collect live leaves from the canopy. We collected tree water by coring into the tree and removing stems from it. Stem core, sorry. And we collected water from the stream, precipitation, groundwater, stream precipitation and groundwater to represent kind of the different sources of water during the summer. And very quickly to understand the nitrogen, we use these really fancy machines that essentially you take the soil or the leaves, you burn them using this machine at a very, very high temperature, 1800 degrees Celsius, and that turns the soil into all the different gases from carbon, nitrogen, all the gases that make up that material, and we're able to measure the ratio of those gases using this analyzer. And what we found is, so again, here's this gradient in the soil depth and the nitrogen. We see that we have this kind of characteristic curve where the values shift dramatically from 0 to 10 centimeters, and then they kind of stabilize as you get deeper. And if we measure the ratio of nitrogen for the maple, oak, and pine leaves, we can match that up to where they fall on the curve to get somewhat of an estimate of where the plants are taking up their nutrients. Excuse me. And what we see is the maple, oaks, and pines are taking up nitrogen from the top 10 centimeters, from relatively the top 5 centimeters of soil, but definitely from the shallowest layers where we see this shift in nitrogen values. But now I'm going to switch to show the water. We collected water from the stems and the soils again, and we extract it using liquid nitrogen and a vacuum pump. And this process is shown by here, where we take the material and we pull all the water out of it using boiling water, liquid nitrogen, and a pump. And we measure the two isotopes, the stable isotope ratios of water, delta deuterium and delta O18. And here is the results for the soil water oxygen. So, water is made up of hydrogen and oxygen. So we're measuring just the ratio of the oxygen stable isotopes in the water here. And we see soil depth is on the y-axis from 0 to 100. We see that there's a characteristic decline of this ratio value as you get down in depth, which is what we need to try to understand if plants are taking resources from different depths. And the oaks are taking up water from what looks to be deeper depths than the maples and the pines, where maples and the pines have an average value that more matches shallower soil layers, where oaks seem to be able to take up water from deeper soil layers. And if we put this all together, here we have the oxygen, and here we have the hydrogen, that delta deuterium. We measured all the different sources of summer water from the rain, the stream, and the groundwater. And here we have the soil water we extracted. So the gray dots are from 0 to 50 centimeters, and the black dots are deeper than 50 centimeters. And we see they fall kind of along this local meteoric water line. And we compare that to the oak, the maple, and the pine. And again, it looks like the oaks are taking up water from deeper depths. So if we put that together with the nitrogen and water, overall, maple, oaks, and pines are taking up nitrogen from shallower soils. But then it appears that the oaks have the ability to take up water from deeper depths. And when we compare this to the root system distributions, we saw that oaks had an increase in root system biomass and also have an ability to take up water from deeper depths. So some of the takeaways for this chapter is, again, oaks had a major increase in fine root biomass and seemed to be able to take up deep water. Something that I didn't show, but what has been seen in the plots is oaks are also responsible for 64% of total new wood growth since 2015. And this occurred during a couple of severe droughts in that time. But all the trees shifted their root system extends towards deeper soil layers when experiencing drought stress. So I'd like to acknowledge everyone at UNH and Stanford that helped with this experiment, and the stable isotope labs that have run the data, and all genetics for the DNA barcoding analysis. So those are my 3 chapters of the PhD, and I'm just going to go through a couple of the quick takeaways. If we bring this back to the beginning, remember that scientists walking around the forest, looking at the canopies, trying to figure out what to do. Well, with some of the analysis we've done, such as building the largest database of root system dimension and volume, finding ways to predict the size and shape of plant root systems based on shoot size, and unearthing some of the interactions between roots and the climate, resources, and neighboring plants, we hope that we can use some of the information about the size and shape of plants above ground and its climate and environment to better understand what's happening below ground. And the big takeaways are that roots show considerable variation in their depth and spread. Plants take up much more space below ground, and we can use allometry to understand the relationships between above and below ground plant size. And trees have an ability to kind of, or we saw that the root systems of trees shifted towards deeper soil layers when facing drought stress. But trees did display kind of various resource acquisition and rooting strategies. And so with that, I'd really like to thank you all for taking the time to learn about my research at Stanford. And I'd like to acknowledge some of the support from the National Academy of Sciences Ford Foundation Fellowship and the National Science Foundation GRFP. This has been a really amazing adventure, one of the most difficult and most fun things I've done in my life. And I'm more excited about my science today than ever. I look forward to continuing in this journey. And please stick around. We're going to do a Q&A, and then we will have some quick acknowledgements after that. So thank you all so much.